Hey, Hammy here, back with part 2 of 4.3 Powering the Cell on cellular respiration. In the first part 1, we looked at glycolysis, the first step of breaking down glucose. In this video, we want to look at the next main step in breaking down this glucose. Okay. So remember, glycolysis, which we talked about in the last video, glycolysis. Just is the first step in breaking down the glucose. It happens in the cytoplasm of the cell. Scientists think glycolysis evolved before these next stages that we're going to study, because glycolysis does not require oxygen. Glycolysis itself, the first step is anaerobic. That can take place whether or not oxygen is even there. It does not require oxygen. Okay, these next couple steps we're going to talk about are the aerobic steps of cellular respiration, and it's going to require oxygen. Okay, so glycolysis is anaerobic, and then the next steps are aerobic. We think glycolysis happened first before the Earth's early atmosphere didn't have a lot of oxygen. And it's a way that organisms got to ATP, got a little bit of energy out of the sugar molecules that they had. Okay, the oxygen wasn't even available until photosynthetic bacteria started to take over and put oxygen into the atmosphere. And as the oxygen in the atmosphere started to build up, we think that organisms evolved kind of these later steps to get more energy out of the glucose molecule. Most organisms today prefer to use cellular respiration because you get more ATP. You get 38 ATP versus just the 2 net gain you get from glycolysis. So we'll kind of take a look at what's the next steps after glycolysis. First, we kind of need to take a look at the organelle where the next steps will take place. Okay, the first step, glycolysis, remember, is in the cytoplasm in the cell. The next steps take place in the mitochondria. Okay, the basic structure of the mitochondria, and you'll see this again, just like you did the chloroplast on your last test. Okay, and like the chloroplast, because of the endosymbiotic theory, we think they were once engulfed by an early eukaryote. It has an outer membrane from being engulfed, and then also its original membrane or inner membrane. So it has an outer, an inner membrane. Okay, the clear, kind of the blue part here, bluish green turquoise part that you see, the fluidy part we call the matrix. This would be similar to the stroma in a chloroplast. Okay, in between the inner membrane, this inner membrane and outer membrane, there's actually a space. We call this the inner membrane space. Okay, when we get to electron transport chain, this space will become important. Okay, and then these little folds, the inner membrane doesn't just go all the way around the inside like the chloroplast did. It actually has these little folds in it to increase the surface area. These little folds we call cristae. Okay, and the purpose of the cristae is to increase the surface area, okay, for some of these processes that we're going to talk about. Okay, so here's the general structure of the mitochondria. And the next couple of stages, I'm going to refer to where these different steps take place. Okay, the first thing that happens, you remember in glycolysis, you had the glucose, and it was broke into, the 6-carbon thing was broke into 2 pyruvates or pyruvic acids, which had, pyruvic acid has got 3 carbons. Okay, so you see it down here, pyruvic acid. This is coming from glycolysis. The first thing that happens in a lot of old books, this was often called the transition reaction. A lot of the newer books our last edition and our new book here will oftentimes just lump this right in with the actual kind of circular cycle. They put this first part in with the Krebs cycle, but it used to be kind of sometimes considered separate. So the first step, you're going to take that pyruvic acid, okay, you're going to break off a carbon in the form of carbon dioxide, okay, and when you do that, when you break a carbon off, you're going to get some high energy electrons. You're going to harness some of that energy in the form of high energy electrons, which is going to be picked up by NADP or NAD plus, and it'll be made into NADH. Okay, this is our kind of our electron dump truck, our full electrons. That leaves you with a 2-carbon substance called acetyl. The acetyl group is connected to a coenzyme called coenzyme A. So we often just call it CoA or abbreviate it CoA. So the pyruvic acid, a carbon breaks off, a CO2, which releases electrons to NADH, and you end up with this little acetyl-CoA, a 2-carbon substance, acetyl-CoA, which is then taken into as it's being taken into the mitochondria and enters the Krebs cycle. Okay, the first step of beginning to break down this acetyl-CoA in the Krebs cycle, which, by the way, is named after Hans Krebs, who helped discover this cycle and all these different steps. The first step is the acetyl-CoA comes in, the coenzyme A releases, and it takes that 2-carbon acetyl group and combines it with a 4-carbon substance, oxaloacetate. Okay, so pause the video or take a moment, make sure you spell that right. Okay, oxaloacetate, which we often just abbreviate OAA, oxaloacetate. So when you have 4 carbons combining with 2 carbons, you get 1, 2, 3, 4, 5, 6. Okay, so you get 6-carbon substance. That 6-carbon substance is called citric acid. Sometimes the Krebs cycle is also called the citric acid cycle because the first substance of the cycle that we recognize is citric acid. Okay, so the citric acid cycle. Okay, and the next step you have, here's our citric acid. The next steps, actually, your book just calls this step 2. We say it's slowly broken down. Because remember, if you break glucose all at once, or you break a bunch of bonds all at once, whoosh, you're going to get this rush of energy, and a lot of it is going to be lost. Okay, so it's broken down slowly in steps to try to capture as much of that energy as possible. That energy is going to be captured as, what do you see off to the side here? The electrons, high energy electrons that come flying off, are going to be caught as, NADH and FADH2. Okay, NAD and FAD are going to pick up these electrons, and we're going to end up taking them to the electron transport chain, which we'll talk about in the next video. Okay, but we make a bunch, and notice we made some NADH up here in our transition reaction, but this book considers that part of the Krebs cycle as well. So we get a bunch of NADH, FADH, and also down here, without doing electron transport chain, it's called substrate level phosphorylation. There's an enzyme that will take the energy and make ATP, okay, which is our energy dump truck. All right, so we have high energy electrons and energy itself. We charged our little battery here. The waste products, the carbons, okay, the carbons in that acetyl-CoA are going to be released as, same thing as the transition reaction, carbon dioxide. Okay, so we're getting energy and we're getting carbon dioxide. Notice what we end up with here at the end of the cycle is our OAA, or oxaloacetate. We have our 4-carbon substance, so it can combine with another acetyl-CoA, and this thing can just be spinning constantly, breaking off CO2 and capturing electrons, making NADH, FADH2, and also some energy in the form of ATP. If we look at our totals, okay, remember one glucose in glycolysis was split, so times 2, into 2 pyruvic acids. Glucose had 6 carbons. Now you have a 3-carbon substance. So how many turns is this whole thing going to make? That's right. It's going to make 2 turns, okay, times 2 per glucose. So looking at that, how many carbon dioxides are we going to get out of one glucose molecule? Well, let's circle them. Here's 1, 2, 3. But remember, this happens twice. So 3 times 2, we're going to get 6 carbons, which makes sense because glucose is C6H12O6. Right there, 6 carbons. Those 6 carbons, okay, become the 6 carbon dioxides that are released. How many ATP? Well, let's count. Okay, we've got 1 down here, but it turns twice, so you have 2. Plus, if you include the 2 that were made from glycolysis, gives us a total of 4 ATP. As far as energy that our cells can use, the high energy electrons now, let's count. Here's NADH, 1, 2, 3, 4. Okay, now this spins twice, so 4 times 2 is 8. Plus, you remember we had 2 from glycolysis, gives us per glucose 10 NADH. And these numbers will be important when we get to the next step and we look at how much total energy does the glucose give us. FADH2, okay, we've got 1 here, and this happens twice, so that gives us 2 FADH. Okay, our cells can use this ATP directly, that was made directly from glycolysis, and down here at the bottom of the Krebs cycle, okay, the NADH and the FADH2, those high energy electrons, are going to be harnessed in stage 3, okay, the electron transport chain, which is going to be similar to what happens in photosynthesis, where these electrons are going to be used to make even more ATP for our cell to use. Okay? So the next video, we'll take a closer look at the electron transport chain and how exactly that is done.